good afternoon, or good evening according to the time you are watching this video. Today I'll be talking about enzymes, a very important topic for both boards and NEET exam. Let's talk about this. Before starting the topic of enzymes, I would like to recall from your lower classes that you guys have studied catalyst in physical science or chemistry section, I guess. Catalyst was something which can increase or decrease the velocity or speed of a reaction and remains unchanged after a reaction. So you can think catalyst is a helper molecule to a chemical reaction. Like that, we have enzymes in our biology. So enzymes are nothing but biocatalyst molecules. So what is the definition of enzymes? Enzymes are proteinaceous biocatalyst which can increase or decrease the velocity. Velocity means speed. The velocity or speed of a reaction and remains unchanged after a reaction. And remains unchanged after a reaction. That's it. Enzymes are proteinaceous molecules, meaning they are made of protein molecules. In fact, all the enzymes we find in biological system are made of proteins. There are some exceptions to this, I'll come to that point later, and they are nothing but biocatalysts which can increase or decrease the speed of a reaction and remains unchanged after a reaction. So I told you guys that all the enzymes are made of proteins, but there are few enzymes. There are few enzymes which are not made of proteins. They are made of RNA molecules. Yes, you guys will be surprised to know that sometimes RNA molecule behave like enzymes, and we call those enzymes as ribozyme. You see the word is ending with the suffix zyme, meaning that it's an enzyme. So RNA molecule acting as enzyme is called ribozyme. We have many examples like RNA speed, peptide transferase. You will see this kind of enzymes during protein translation. So we'll come back to the original topic. So enzymes are biocatalysts. Now you have to know some basic terminologies regarding enzymes like substrate, product. Let's take a reaction actually. Perhaps you guys have studied this reaction in your digestion chapter in class 10th or 11th standard. That is, in your mouth, the boiled starch, that means the rice you eat actually, that contains boiled starch, is broken down into maltose by the enzyme called ptyalin or salivary amylase. Yes. Ptyalin and salivary amylase both means the same. So boiled starch that you eat regularly, that is converted into or broken down into maltose. Maltose is a disaccharide containing 2 glucose. So here boiled starch is the substrate. This is the substrate. Ptyalin, this is the enzyme, and maltose is the product. So substrate means basically reactive. So an enzyme acts on a substrate and converts it into a product. Yes. Enzyme acting on a substrate and converting it into a product. That is the thing about substrate and product. Very simple thing, I hope you guys understand easily. Now very important topic for your exam. That is enzyme classification. Yes. IUB, this international body, meaning International Union of Biochemistry. International Union of Biochemistry. They have divided enzymes into 6 classes. According to your NCERT, it is 6 classes, but nowadays it is now 7 classes actually. I'll talk about that later. So, according to NCERT, I'm going on. IUB has divided all the enzymes into 6 classes. And you can remember these 6 classes by the simple mnemonic OTHLIL. OTHLIL, OTHLIL. Each letter stands for each class of enzyme. So O stands for oxidoreductase. T stands for transferase. I'm going to explain each and every type of enzyme very soon. Before that, just know the full name. O stands for oxidoreductase. T stands for transferase. H stands for hydrolase. L stands for lipase. I stands for isomerase. And there is another L that is for lipase. So, all the enzymes you find are divided into 6 groups or classes: oxidoreductase, transferase, hydrolase, lipase, isomerase, and lipase. 6 classes. And you can easily remember the 6 classes by using this word OTHLIL. And actually you have to remember these things serially. Because when you say class 1 enzyme, you mean oxidoreductase group of enzymes. When you say class 3 enzymes, you mean hydrolase group of enzymes. So let's start discussing first class of enzyme, oxidoreductase. So first class, okay. So this is the class number, class 1 or group 1. Class 1 enzyme is your oxidoreductase. Oxidoreductase. So I think you guys can easily understand from the name itself that this type of enzyme does the job of oxidation reduction in a reaction. Let's say S is a substrate, S dash is another substrate which is converted into, let's say S is been oxidized here and S dash is reduced here. So if S is oxidized, that means previously it was in reduced condition, and if S dash is reduced, that means S dash was previously in oxidized condition. So yes, S has been oxidized and another substrate S dash has been reduced. You know from your basic chemistry knowledge that oxidation reduction happens simultaneously. It's not like that oxidation and reduction are independent, because you know that oxidation is removal of electron and reduction means acceptance of that electron. So if somebody gives our electron, there should be someone who takes that electron. So oxidation reduction happens as a couple. Okay, they always go hand in hand. So that's why this type of enzyme does the job of oxidation reduction, that is redox in a reaction. One substrate is oxidized and another is reduced. So that is the way of oxidation reduction, oxidoreductase enzyme mechanism. So I'll give you some authentic examples, like pyruvate dehydrogenase. Pyruvate dehydrogenase. You will find this enzyme during aerobic respiration. This enzyme does a reaction which links your glycolysis and Krebs cycle, ATP yielding process, energy producing process, pyruvate dehydrogenase. Let's look into their name. This enzyme says dehydrogenase. Dehydrogenase, D means not or removal. Dehydrogenase means there is removal of hydrogen. Now, from a basic knowledge of chemistry, you recall the thing that what is this thing called? The removal of hydrogen. This is called what? Think about it. Oxidation. So there are actually 4 concepts of oxidation reduction. You can write like this. I'll write in short. This means oxidation and this means reduction. So addition of oxygen is called oxidation and removal of oxygen, reduction. Okay. Then removal of hydrogen is called oxidation and addition of hydrogen is called reduction. Removal of electron is called oxidation and addition of electron is called reduction. And one more concept is there actually, oxygen number. This concept is taught in class 11 redox chapter in chemistry. If oxygen number increases, we say it as oxidation, and if it reduces, we say it as reduction. So pyruvate dehydrogenase. Just look into the word dehydrogenase, meaning it is removing hydrogen from some substrate. So it is a kind of oxidoreductase enzyme. I'll show you the actual reaction. So there is a substrate called pyruvate or pyruvic acid, same thing, which is a 3-carbon compound which is produced after glycolysis, at the end of glycolysis. You start this thing in aerobic respiration. So I'm taking that example for the time being. So pyruvate is converted into acetyl-CoA, which is having 2 carbons. And in this process, a carbon is lost as carbon dioxide, and also CoA comes, coenzyme A comes. Coenzyme is a big molecule. Don't think think it like a guest. CoA will come and go away. It has just come here to help the reaction. And another thing, NAD plus is converted to NADH. NADH plus H plus. So you understand this reaction. Pyruvate, this substrate is converted to acetyl-CoA. And here is removal of hydrogen actually. How? Here you see NAD plus is removing some hydrogen from pyruvate and itself becoming NADH2, which is actually NADH plus H plus. And here the enzyme is pyruvate dehydrogenase. The enzyme acting here is pyruvate dehydrogenase. So this enzyme is removing hydrogen from the pyruvate. And who is helping in this process? NAD plus. This is nicotinamide adenine dinucleotide. We'll start this thing under the cofactors section of enzymes later on. So I hope you guys have understood this thing, oxidoreductase enzyme. So oxidoreductase enzyme means they do the job of oxidation reduction in a reaction like this. This is called a link reaction. This is the link between glycolysis and Krebs cycle. Then class 2 enzymes. OT, T stands for transferase. The second class or group of enzymes is called transferase. So second class enzymes, not second class, same class of enzymes, class 2 enzymes are called transferase. Transferase class of enzymes, they do actually transfer one certain group from one compound to another compound. Let's take an example. Let's say S bond G, SG. This is a compound. G here stands for a group. It is reacting with another substrate S dash. Now, after the reaction, what is formed? S plus S dash G. You see this skeletal reaction, I have not drawn, I have not written an actual reaction, I have shown you guys a skeletal reaction. So SG plus S dash is producing S plus S dash G. That means this group G is transferred from this S to S dash. This group G has been transferred from S to S dash. So which type of enzyme is acting here? Transferase. Meaning a group is transferred from someone to another. You can take some authentic example. I'll take the first reaction of glycolysis for this purpose. We say glucose is converted to glucose 6-phosphate. Yes. I'll take the first reaction of glycolysis. Glucose to glucose 6-phosphate. Before diving deep into this thing, I want you guys to understand one thing, that glucose 6-phosphate, how many phosphate does it contain? I'll give you 5 seconds to think about it. Glucose 6-phosphate, how many phosphate does it contain? I hope you guys have thought about this thing. If your answer is 6, you're wrong. If your answer is 1, then it's correct. Glucose 6-phosphate does not mean that it has 6 phosphate. Think about IPS nomenclature. Glucose 6-phosphate means there is 1 phosphate which is attached to the 6th carbon of the glucose. You know, glucose is a 6-carbon compound having the formula C6H12O6. So glucose 6-phosphate is having a single phosphate attached to the 6th carbon of the glucose. So glucose is converted to glucose 6-phosphate. And here ATP is helping in this reaction. ATP comes and it goes away as ADP. You know ATP? ATP is your energy currency known, the full expansion of this is adenosine triphosphate, and ADP means adenosine diphosphate, meaning ATP has 3 phosphate in it and ADP has 2 phosphate in it. So ATP is giving 1 phosphate to this glucose, so that glucose becomes glucose 6-phosphate and ADP triphosphate becomes diphosphate. So there is a transfer of phosphate from this ATP to glucose. There is a transfer of phosphate from this ATP to glucose. So this is the enzyme acting here is a kind of transferase enzyme. And the name of the example, enzyme is hexokinase. Hexokinase or glucokinase, you can say. Both does the same job actually. Hexokinase or glucokinase. So what name is like this? Hexokinase you say, or glucokinase, let's take the example, glucokinase, this name. The enzyme's name are made in such a way that it reflects its action. So glucokinase, it is acting on the glucose. So first part of the name of this enzyme has been taken from the name of the substrate itself. So from glucose we have taken the portion gluco. And kinase, kinetic, it's relevant. So kinetic means movement. So movement means transfer. So yes, glucokinase is a transferase type of enzyme which is actually transferring 1 phosphate from ATP to glucose. And in this process, glucose becomes glucose 6-phosphate and ATP becomes ADP. So 1 phosphate has been transferred from ATP to glucose. So glucokinase is a transferase type of enzyme. It's an example of transferase. And glucokinase is also called hexokinase for the reason that glucose is a 6-carbon sugar and it is also known as hexose. That's why glucokinase is also called hexokinase. So this was the second class of enzyme, transferase. The next class of enzyme are very easy. OTH, meaning H for hydrolase. H stands for hydrolase. What does hydrolase do? Simple. They split a compound using water. They split a compound using water. Let's take a binary compound AB, and water we can write HOH. H2 we can write HOH obviously, and it will be converted into AH and BOH. So AH plus BOH. So you see this compound AB, A bond B, is broken down into 2 parts using water molecule. And this is done by which class of or which type of enzymes? Hydrolase. And this is happening in a body ample number of times actually. Like we do eat 4 to 5 times a day. So digestion is occurring in our mouth, stomach, and intestine, also small intestine. And all those digestive enzymes are actually hydrolase type of enzyme. Let me give you some authentic example. You know, in your small intestine, in small intestine, what is happening? Maltose, which is a disaccharide molecule. Maltose is broken down by the enzyme called maltase. You know, the name of the enzymes end with ASE. The maltose is broken down into 2-glucose by the maltase enzyme. And this is actually a hydrolytic process. Hydrolase enzyme is required for this. This maltase you see here is a hydrolase type of enzymes. I'll do the reaction. Maltose, the formula for maltose is C12H22O11. And I hope you guys are smart. This looks surprisingly like the formula of sugar cane sugar. Yes, same formula as maltose. Maltose is the same formula as cane sugar, C12H22O11. Add some water and break down by 2. C12H24 and O12. Break it into 2. Same thing, right? So maltose is broken down into 2-glucose by the action of which enzyme? Maltase in small intestine. And it is a hydrolytic process. So maltase is a hydrolase type of enzyme. In fact, all the digestive enzymes you have in your salivary juice, like tal in your salivary amylase, or pepsin in your stomach, or sucrase, maltase, pancreatic amylase, trypsin, all those digestive enzymes are hydrolase in nature. They are all examples of hydrolase. So very important. It's hydrolase type of enzymes are required in your digestion process every day. So this was the third type of enzymes. Now, class 4. Class 4 is something which also breaks down a compound, but unlike the hydrolase, it does not actually use water. So class 4 is called lyase. It also breaks down a compound, but without using water. Let's take an example. C bond C. Okay. Let's do a skeleton reaction. Let's say some atom or element X attached to this carbon and Y. And we are not caring about other values of carbon. Okay. So let's say this is a compound. Now, X and Y, they form a bond and goes away together. So if X and Y joins hand and leaves away, then what is happening? This carbon will withdraw this bond. So this carbon, these two carbons will form a double bond. So don't think about this two. Any group may be there. I'm just drawing a skeleton reaction. So CX will C bond with C. X is attached to C and this Y is attached to this carbon. And X and Y are going away together, joining hand in hand. So this carbon will form a double bond in this way. So here you see this compound is broken down into two parts, X bond Y. This is one part, and another part is this. This compound is broken down into two parts, but without using any water. So this is the action of lyase group of enzymes. So lyase, any lyase enzyme breaks down a compound into parts, but without using water. Just look at this example. X and Y goes away together, and this remains. And once need to ask this question: which type of enzyme, after doing its action, leaves a double bond in the product? You see, in the substrate here, in the substrate there is no double bond. But after the reaction, a double bond has been formed in this compound, right? So it creates double bond. Which type of enzyme creates double bond in the product? Lyase. So this was the fourth class of enzyme. And then fifth class of enzyme, that is your isomers. Isomer means compounds which have the same formula but different structure. This is a formula for glucose as well as fructose as well as galactose. So what is the difference between them? Glucose is having aldehyde group, galactose is also having this aldehyde group, but fructose is having ketone group. So isomer. Isomer means compounds having same molecular formula but different chemical structure. Compounds having same molecular formula but different chemical structure. Those compounds are called isomer. For example, glucose and fructose are isomers because glucose is having this aldehyde group and fructose is having this ketone group. We will deal with isomer more in the chemistry section in organic chemistry. So isomers. So what does this type of enzyme do? This type of enzymes converts the compound into its isomer. Let me give you an example. I will take another example from glycolysis. So glucose 6-phosphate is converted into fructose 6-phosphate. Let me tell you guys, don't think about this phosphate. Here the phosphate was attached to the 6th carbon of the glucose, and here also. Phosphate is attached to the 6th carbon of the fructose. But the thing is that glucose is converted to fructose. So glucose is converted to its isomer, which is fructose here. So which type of enzyme is acting here? Isomerase. And the full name for this enzyme is phosphohexoisomerase. Yes, phosphohexoisomerase. If you want to write full name. So phosphohexoisomerase is converting this glucose 6-phosphate to fructose 6-phosphate, and the enzyme does the job of isomerization. That means glucose is converted to fructose. And the last class of enzymes, class 6, that is ligase. Before talking about this ligase, I want to tell you guys something interesting, something noteworthy. You have seen many kinds of enzymes till far, but you see one common thing, that most of the enzymes are breaking down compounds. Most of the enzymes are breaking down compounds. So, isn't there any enzymes which join compounds? Yes, there are, which join compounds, and those enzymes which join compounds are called ligase. Ligase comes from the word ligation, means joining. So ligase group of enzymes join two molecules. Let's take a famous example, DNA ligase. The name of the enzyme is itself DNA ligase. DNA ligase is known as molecular glue. DNA ligase is known as molecular glue because it is joining fragments of DNA together. Like this. Yes, DNA ligase is joining the fragments of DNA together, and this enzyme is known as molecular glue in the field of molecular biology or biotechnology. Very famous question, this DNA ligase. So this is another class of enzyme, ligase. So these 6 type of enzymes were given in NCERT, but in recent classification, there has been another type of enzyme. So 6 class of enzymes have been given in NCERT, but there is a recent addition to this classification by IB, which is class 7. It is translocase group of enzymes. Translocase group of enzymes are associated with transfer of molecules or ions from one side to another. Let me give you some example. The famous example for this group of enzymes is ATP synthase. ATP synthase, you will find this enzyme in the mitochondria. It is present in that F0 particle actually, and it is associated with production of ATP, which you will learn later in ETS. But this enzyme is associated with hydrogen ion movement. Okay, there is a thing like this. Let's say this is your mitochondria. This is your absorption particle situated on the inner mitochondrial membrane, and this enzyme uses the hydrogen ion gradient which is flowing from the perimitochondrial space to matrix to produce ATP. You will see this enzyme in detailed action in the topic called chemiosmotic theory. It has been there in your photosynthesis and your respiration chapter. So for today, it is the end. I'll come back with new topics later on. Stay healthy and keep studying.